killed like a third of the, or more of the European population. But after that, you start to see that we enter into an exponential increase that has been sustained for at least a couple hundred years, but now we are entering a slowdown. If we zoom in this last portion of this graph, what you are going to observe is that the peak of increase was reached in the 60s when we were having 2.2% increase in the human population, okay? More than double, because usually a replacement for one single human is to produce on average 2.1 humans. So if you have two babies, you're doing great, you're replacing your own in the population. If you have less, the population still is adding, but it's not, it might not be increasing. It might just be replacing a few humans, but most likely is going to start to decrease, okay? But then, later on in the 60s, you start to see that from this peak, we started to have a lower percentage of increase. We are still adding humans, a lot of them, okay? And it is projected by the middle of this century, we are going to be reaching 9.8 billion people on the planet. But of course, you know that resources are limited, as we've seen in many examples. That also applies to humans, but of course, we are always eager to rely a lot in our ingenuity in order to produce the food that we need to sustain a growing population. Okay, again, we are no longer in full exponential growth. We are still adding people, but that is slowing down, as you can see in these lines, and the projection is that, yeah, human population is going to keep increasing, but you can start to see that there is like a slowdown in the growth rate of the human population for the future, and the rate of growth, you can start to see that is going down. Okay, that makes sense. So we are adding less and less and less people on average per year. So there are one important concept in all this about population ecology and in demography is called demographic transition. So basically it's how the individuals that are in one stage of the life cycle or in an age class are going to survive and move into the next stage in the demographic cycle. And that is important because when you start to analyze demographic cycles, you can extract information that is going to tell you if that population is likely to grow or is a population that is in decay, because there are more older people than younger people. And once we extract this information, what we can produce are graphs that look like this, okay? So a graph that is telling you of a population dominated mostly by young, productive people is going to look like this. So less older people in the population, and the base of the pyramid is thicker. But then when you start to see the opposite direction where the population is slowing down or actually not growing at all, you start to see something like what is happening here in the US, or which is slowing down, because now you can see that the pyramid doesn't look like a pyramid anymore. The older population is started to get thicker compared to the younger population that is the economically sustainable population for all the rest of us. Okay, so this is a slowdown typical graph of demographic transitions. And then no growth, like in many countries in Europe, this is one case, is Italy, but most of the European countries you are going to see something like this, or even more dramatic in some of them where the pyramid is actually inverted. It's thicker on the top than at the bottom, and that's very, very bad news for anybody in that country. Of course, these diagrams hide a lot of information behind them that is very important for demographers and population ecologists. For example, a population like Zambia, or that represents many of the developing nations around the world, where many people are having lots of babies at young ages, what is going to signal at somehow is that there might be a lot of unemployment, not enough opportunities. So because there is more and more addition to these younger age classes in the population, but there is not enough productive or economic production of jobs that are needed to be filled out. And also that leads to also understanding cultural differences and the opportunities that people have for a good living. Because in addition to that, yeah, they are producing more, but death rates are usually higher compared to developed nations. Now, if you compare United States and Italy, what you are starting to see is that a lot of the population is retiring, like the boomers, and then the younger generations, because of constraints of the urban environments, are deciding to have less babies. Why? Because there is competition for jobs, there is a lot of competition for trying, quote unquote, being the best at whatever you do and the only thing that you do. So all these narratives that we turn out to believe, which are actually more damaging to us as individuals than actually they are to the country. Okay, so all these narratives of a social species like us are going to have a strong impact on the outlook of life for younger generations, like yourselves. Probably you have heard of the issue that probably Social Security is not longer going to be viable to sustain anybody else, especially anybody in this room, which is a lot younger than me. So that's why you have to plan better for the future and have babies. Babies are the future. Have more babies. Don't trade your family for career things, okay? That's based on experience, that's not the best way to go, okay? But of course, it's an individual decision, but better to have more babies. Okay, that will lead us also to a situation, right? The carrying capacity concept that we were talking about earlier. So the planet is just one. Resources are right there, and if we don't get new methodologies in order to extract more resources to feed a growing population, we are going to get in trouble. So we first have to handle what is the carrying capacity of the planet, or how many humans can the planet sustain. And the range has gone from just 1 billion, that we are way past that number, all the way to 1 trillion, which is ridiculous. Those are two extremes, but who knows, right? So there are ways that we need to consider in order to establish what is the carrying capacity of the planet. And when people, researchers, economists, and other social scientists and environmental scientists have considered many of the factors like food resources, housing, energy per individual, depending where you are on the globe is going to be the energy that you require, because culture is different across the planet, so the demand for different food needs or different energetic resources are going to be different depending where you are living on the planet. But if we average everything around the planet, there is a certain amount that every person needs in order to have a stable and let's say more or less a stable life, to cover all the basic needs. But when we compare those numbers to what actually people are using around the globe, you can see that things are not looking that good. So the per capita ecological footprint at global hectares per person, so we don't have insufficient data for those places, which are basically where almost nobody's living. And we have this data showing that usually developed nations, the ones in warmer colors, some in Europe, in North America, Australia, are taking up a lot more land area of what they are supposed to have. So more than 6 hectares. So everybody should be around using what they need. So when you standardize the units, it should be like the same land mass per person, but you can see that developed nations are using more resources than they should be using compared to other nations, okay? So that's the disparity that we have around the world, and something that we need to care about if we are to have a sustainable future for everyone. Another way to look at the differences in energy uses, and energy means every type of resource that we need to sustain our lives, can be in terms of showing energy, how energy is used. And this map, which is very famous, you can see the energy demands, right? How much light is used in different parts of the globe. And that gives you an idea of the demands that we are imposing on the planet on different parts of the world. Okay? Any questions? No? Ten more seconds. Okay. All right, so that's for the chapter on population ecology. Let's move now to community ecology. And before I start this chapter, for next week that we have the exams, both exams are in person, and we are going to follow the same way that we did the last exam. So before you leave the room, you have to show your ID and that you have closed the exam to your TA. And I sent an announcement this morning how I'm going to handle the last exam. Those who were dishonest, that is going to be the exam that is going to be dropped out, is going to be your lowest grade. And so hopefully for the remaining two exams, you are trying to be honest. And remember, for those exams, only laptops are allowed. No privacy screens or anything like that. If you don't have a laptop, find one, borrow one from the library, borrow one from your friend. Only laptops. If you don't bring a laptop, you automatically have 0 on the exam. And yeah, I think that's all I have in terms of exam on Tuesday is going to include all the way to environmental ecology. So hopefully we can cover all the community ecology today, and you will have to study on your own. You have the PowerPoint for the next chapter, and then the final exam is going to be basically everything, okay? So it's going to include also the last chapter on conservation biology, so you will practice with your homework. And also, if you have questions or you want to ask something, you have your TA, Eli, and also myself. So just send us an email. We would be happy to help. Yes?
So does exam 4 count chapter 56, or is it only the final?
No, only the final for 56, which is conservation biology. For Tuesday is community ecology, and the next one, 55. Okay? Ecosystem ecology. Okay? Or I don't know if it is called ecosystem or environmental ecology, but yeah, but the next chapter after this.
In the beginning of the year, you mentioned how we could have 5 sheets of paper for the final
Yes.
exam. So for the last exam, you took the third one. You said we could use 2 pieces of paper.
Yeah, thank you for bringing that up. For next exam, because I'm not covering all the material for the next exam, you can also bring 2, okay? So you can make your...
Those 2 pieces of paper, I'm sorry to interrupt, would those 2 pieces of paper also be used in the final, or can we only
No, for the final, you can use all the ones that you already prepared from the beginning of the semester. Okay, so if you have them, those are the ones that you can use.
Okay,
Okay?
thank you.
Everyone is clear? Yeah?
Is the final like a combination of the previous exams, or like what do you think is like most important for us to focus on?
I would have to check because I have many questions on the question bank and Canvas, I set up Canvas to pick randomly. So it's going to be basically everything. So it can be from any chapter
Okay,
for the final.
alright.
But that's why I'm saying that whatever notes you prepare for previous exams, those are the ones that you can bring. Okay? All right. And again, so don't get bogged down by details about very specific things that we cover in the semester. Okay? I am more interested into you understanding concepts and making connections across concepts. So if you have seen many of the questions that you have to answer implies that you understand the concept that you are asked about and that you are also able to make connections across concepts from different topics. Okay? So that's the most important skill that you have to develop. Don't try to learn things by rote or to put as many details that are really unimportant on a cheat sheet, but rather focus on understanding the concepts. And nowadays with the textbooks that you have, that is even easier because the same textbook at the beginning of the chapter highlights what are the main concepts that you need to understand. Okay, and how do you understand them? By making connections across different topics, which is, if you realize, that's what I've been trying to do with you across the whole semester. When I am asking you questions about some topic, you also have to learn about or remember the other topics that we cover in different chapters to be able to answer that question. That is a more important skill than trying to remember every detail of every single thing. Okay? Clear now? All right. So community ecology. Across the semester, we've been indirectly mentioning many things about community ecology, in particular about ecological interactions, okay? Here is an example of a mutualistic interaction of this fish that basically removes ectoparasites from the mouth of this eel, and so both get benefit, right? Get food and one gets clean. So we are going to be talking about interactions like this. We are going to go into more detail. And also, we will go into some other important concepts in terms of community and also related to ecosystem ecology, like what a foundational species is, what disturbances are important to be understood in terms of ecological interactions, also what a keystone species is. It's not the same as a foundational species, so we will learn that. Let's start by basic concepts about communities. So it is not here, but it is in your textbook. What is the definition of a community? Is a group of populations of different species that are interacting among each other in the same space and in the same time. So basically you are just moving one rung up from the population level to the community level. Population level, you are talking about individuals of a single species. When you move to communities, you are talking about individuals of many different species, okay? And when we talk about community structure, what we are trying to do is to give an idea or define how the community is built. Like for example, by composition, we mean what species are in that community. What is the composition of a species? What do you find in there? The relative abundances. And then also, we want to understand the abundance structure, not only saying there is this number of species. The abundance structure is going to tell you what species are rare and what species are common or dominant in the community. All those things are going to be very important to understand how the community is structured. And of course, interspecific interactions, we have already mentioned these ones. I'm not going to stop there. We are going to go into more detail. And the way we code them is just with symbols. Positive, positive refers to a mutualistic interaction where all the involved organisms benefit from each other. Positive, negative is like parasitic interactions. Zero and a negative and a positive symbol is like commensalism. What one gets benefit, the other really doesn't care. There is no positive or negative effect. When we talk about competition, it is a negative interaction because both species are going to be competing. And usually what species are competing for is food resources or some type of resource that can be limited in the environment. That's why they are competing for it. A species will never compete for resources that are highly abundant and everybody can get the fair share that they need in order to continue doing their regular activities. Now, within competition, there is something that is known as competitive exclusion. Competitive exclusion means that when two species engage in competition for a limited resource, one of them is going to be better or more efficient at exploiting that resource, so it's going to outcompete the other. They are not going to be able to live in the same space and at the same time. So that actually is also a force behind the production of new species, and we will mention some cases. And over there in the slide, what you see is a classical example that was in the last century conducted in the lab using paramecium species, these protists, that were put into competition for the limited food resources on petri dishes, and one of them was clearly a winner, excluding the other species. Now, other important term in community ecology and ecology in general is what we call ecological niche. The ecological niche includes all the biotic and abiotic resources and all the environmental conditions that the species needs in order to be viable and successful at reproducing in the location where it is found. Usually something that is not clearly stated in textbooks but also belong to the ecological niche is what is known as the functional niche of the species. By function, meaning what guild the species belong. What is a guild? For example, if you have herbivores, herbivores are not all herbivores belong to the same animal group. For example, you have insect herbivores and you have mammal herbivores. Nothing to do with each other. They diverged from the most recent common ancestor very long ago. But they belong to the same functional group, which is herbivores. That's another part of the niche. Basically, what is the role or the activities that the organism is doing in the environment, okay? And the niche concept can be used to formalize and make the issue of competitive exclusion in a more quantitative fashion, because now with knowing the niche of the species, you can do quantitative estimates of the potential for competitive exclusion when two species come together. Now, competitive exclusion is not that the winner basically is making the other species go, or set of species go extinct. That is a single outcome. Another more common outcome in nature out of competitive exclusion is resource partitioning. Resource partitioning means that, okay, I'm not that good at competing for this resource against this species, but I am able through evolution and through generations to adapt to other resource that is present in the environment, so I don't need to be competing with these other species for this resource. So I adapt and specialize into using other resources in the same environment as these other competitor species. This is one example of how anolis lizards, especially in the Caribbean islands, have adapted to exploit different niches on the same environment. For example, you have these anolis here that actually like to be in lower portions of the forest that it inhabits. but it prefers more open spaces. It likes more the sun compared to other species like this one that usually is higher up on the trees and in more shaded conditions. And you can see here other names of other Anolis species that have adapted to exploit other portions of the habitat that they are living in. And all of these species out of evolutionary studies are known to share a recent common ancestor. So this is a type of evolution that is known as what? Anybody remembers when you have a common ancestor and many species come out of it to exploit different niches in a rapid succession most of the time? Like Darwin finches is another example on islands. You are going there? Radiation, yeah, but there is one word missing. What is the word before radiation? What do you have to do in order to be able to exploit a new resource? You need to develop what through evolution? Adapt, oh, who said? Oh, okay, I didn't hear you, sorry. Adaptive radiation, okay? So this is an example of an adaptive radiation. Okay. Are there any questions? No? More seconds. Okay. Now, when we are talking about the ecological niche, we ecologists have come with a subdivision of the niche that is called fundamental niche and realized niche. The fundamental niche represents the set of conditions, biotic and abiotic conditions, where the organism can be successful, can be able to reproduce and having a good life, okay, so to speak. So all the conditions where that organism can survive and reproduce and have all their needs covered is the fundamental niche. Yet, in reality, what we observe for every species is the realized niche. And the realized niche is going to be that portion of the fundamental niche where you actually find that species. And why is important this difference? Because different ecological interactions like competition or predation are going to limit the geographical distribution of a species. A species can potentially be living in a region of the planet, but maybe it is not there because there is a competitor species, okay? And that competitor is more efficient than the other species and it limits the distribution. So the other species cannot be living there. Or predation or parasitism, right? So if there is a very lethal parasite in a region of the world, then that species cannot colonize it. And there is one very good example for us humans. Like we humans haven't been able to really adapt and live successfully in the Okavango region in Botswana and in that region of Angola and Botswana. And anybody remembers why we mentioned the parasite that is making the conservation case for that region, keeping humans at bay? No, malaria is further up, but we humans have managed to adapt through the sickle cell gene. It's also transmitted by a fly and is a trypanosome. Chagas is in the Americas. It's a cousin of Chagas. Yeah? Sleeping sickness, okay? Trypanosoma brucei. And that bug has made the difference for having a good conservation of all that region in Southwest Africa, mostly Botswana, Angola. Why? Because we humans cannot develop adaptations to withstand that parasite. If you get it, it's 100% sure that you are going to die. So that is one case of showing realized niche. Even though with all our technology, we haven't been able to really settle in that region of the world, with a few exceptions, right? But we humans are not really there, and we cannot do it thanks to an ecological interaction. Here we have an example of barnacles competing for space in the littoral zone, and what researchers did was to show that the fundamental niche of the red species, that is usually restricted to the higher portion of the tidal area in the coastal zone, is just due to competition with the other blue species down there. So what they did was to remove all the blue species from some patches and left others as controls, and sure enough, when there was no competition, the red species extended its distribution in that littoral zone. So that is showing you how the fundamental niche might be different to the realized niche, and most of the time realized niche is not going to be as the same as the fundamental niche. Now, this is talking about the space, but also avoidance of competition can be due in a temporal fashion. For example, when you have these two species of rodents, the common spiny mouse and the golden spiny mouse, they are nocturnal. They prefer to be nocturnal, okay? But when they are found in the same place, then one of them is going to become diurnal, meaning active during the day, and that is going to be the golden mouse. Okay, so you can see there that there are differences in the niche partitioning when they come together in the same locations. One prefers then the daylight instead of competing for resources with the other species of mouse. Of course, this can lead to character displacement. Like in this example of the Darwin finches, you have here the big sizes of these two different Darwin finch species, and you can see that the depth of the beak is not that different between the two species when they are living on different islands. When they are not together on the same island, more or less the beak is the same in terms of depth. But put them together on the same island, and you can see how eventually through a few generations there is what we call character displacement. So big depth is going to be different between those two species because of the resources and the survival differential of the species that carry different depths in the beaks. So you can see that here. When they are not together, you can see that the beak depth is not that different. But together in the same island, Fuliginosa, this species here, the beak depth gets smaller compared to the other one. That is what we call character displacement, and it's an evolutionary response due to ecological interactions. Questions?
How come one doesn't just stay the same, the other changes?
Say it again?
How come one doesn't just stay the same, the other changes? Like, why do they both go to both ends of the spectrum?
Oh, because of the resources that they are going to be competing with. Because remember, they are not the same species and they might not be the same size. So one is going to outcompete the other for the same resources. So the individuals, instead of going into competition with a larger bird, they prefer to go for other resources that doesn't require larger beaks. So there is lower mortality in birds that have a smaller or less, shallower beaks. Okay?
So
Because remember, this is natural selection. So those individuals with larger beaks that are trying to compete with a larger species are going to suffer higher mortality because they are not getting enough resources. But those species that avoid this competition and access other resources with their shallower beaks are going to be more successful at reproducing because they are avoiding this competition. No? Okay. Ten more seconds. Ten more seconds. Okay, now let's move from competition to exploitation interactions, positive, negative. And there we find three different interactions: predation, herbivory, and parasitism. Even though they are coded by the same signs, they are not exactly the same. Because predation kills the prey, parasites do not try to kill the host that they are infecting because that's their food resource, even though sometimes they kill it, okay? And herbivory usually doesn't kill the host plant, okay? So when we are talking about predations, predator-prey interactions, you are going to see the evolution of very fine adaptations. And one example is snakes. Snakes have developed very fine adaptations to find their prey, and they are like a huge sensor in their head. They have olfaction, they have infrared, they have a smell with their tongue. When they are flicking the tongue, they are tasting the smell in the air. All those adaptations are fine-tuned to the prey that they are hunting, but the prey are not just sitting there to be eaten by the prey, but they also develop different types of defenses. For example, mechanical defenses like this porcupine, or chemical defenses like the skunk. And some chemical defenses are obvious to potential predators because they are in bright colors. Bright colors in nature are known as aposematic coloration. Aposematic coloration basically transmits the information to a potential predator. If you eat me, you are going to have a bad time because there are chemicals that are going to damage you, or if not killing you, you are going to have a very bad time, okay? So that's aposematic coloration. Also, many adaptations of prey is in terms of their behavior. They can change their behavior in order to avoid their enemies. In terms of morphology, we have what we call cryptic coloration. Like in this picture, there is a frog in there that is just mixed with the background. So that is a very successful strategy for many potential prey species. But within crypsis or blending of some sorts, we have two types. One is Bayesian mimicry, where a potential prey that is actually palatable for the predator is present in terms of morphology like a species that represents a danger for many other species. Like in this example, what we have here on the left side is the caterpillar of a moth that represents or is very similar to this green snake that has venom, okay? So that helps to avoid predation by these otherwise palatable species like this moth, okay? So that is Bayesian mimicry, where two species that are not related or not closely related to each other look very similar, but one can be eaten and the other is dangerous, is poisonous. Mullerian mimicry is basically the same in terms of the appearance. Both species are going to appear something dangerous, but in this case, both are dangerous. So the predators need to learn to avoid these species, and this is what we call Mullerian mimicry, okay? Questions? No? In terms of behavior, many predators have also evolved behavioral changes in how they present themselves depending on the circumstances. Like this octopus is able to mimic different things by changing the shape so they can either avoid being eaten or to get closer to the prey so that they can suddenly jump onto the prey and consume it. Like this same species you can see here that is mimicking a sea snake to represent a danger to potential predators that would like to eat it. In this case, it's a flounder. It's mimicking a flounder fish, which is basically harmless to many species and potential prey of this octopus, but it mimics itself as a fish, so it can get closer to the prey. Here is a stingray, so it mimics a potential danger for potential predators that consume this organism. Now, other positive negative interaction is herbivory, and herbivores are different to predators because they usually don't kill whatever they eat. They consume just part of the prey, in this case a plant, but they don't kill it. Now, in this case, also, the plants are not just sitting idle waiting to be eaten, either by mammals like this one or by insects, which insects actually represent the most common herbivores on the planet. So plants also will develop some chemicals in their tissues in order to protect themselves and try to deter the potential herbivores. Depending on the plant species, the chemicals can be more toxic or not. And they are known as secondary metabolites. Okay? Now, some chemicals that are not lethal, they can create problems for the development of the embryos of some of the herbivores. Okay, or some of them are even less damaging, they are just distasteful, so that avoids that the herbivore continues to consume it. Then we have parasitism. We have already talked about parasites in other lectures, but basically this case, parasitism, is also different to predation because the parasites don't try to kill the host. They try to keep it alive, quote unquote, to keep it alive for as long as possible because they completely depend on that host for their survival and for their reproduction. Now, parasites can be largely divided into two groups: endoparasites, like worms that people can have in the intestines, or like malaria or other trypanosomes that are circulating in the peripheral blood or are infecting internal organs. And then ectoparasites like ticks, chiggers, mites, okay? Those are examples of ectoparasites. Mosquitoes or blood-sucking insects also are known as parasites, but also known as micropredators because they are free-living, but they relied on some resources of some host species, but it's not that they have to be permanently living as parasites. And one thing that is not mentioned in here is that some groups of organisms undergo what is called hyperparasitism. Hyperparasitism, also known many species as parasitoids, those represent something more similar to predation. A parasitoid is like a wasp species that is going to inject eggs into a caterpillar. And those eggs eventually are going to come out the babies, and the babies are going to consume the caterpillar. So basically they end up killing the host. So that's why parasitoids or hyperparasitism is considered something more similar to predation in ecological terms. Okay? And I already discussed in other lectures about the life cycles of parasites and how important that single thing is for anybody to try to control or to eradicate any parasite that can create any problem that we care about. If you want to control or eradicate a parasite, you need to focus on having a complete life cycle of the species. If you know the complete life cycle of the species, you are better equipped to find the weakest points on the life cycle of that organism in order to try to find a defense to get rid of that organism. Okay? Of course, there are some parasites that are going to have simple life cycles or direct life cycles. They don't need to jump outside of the current host or to a different species. And we have parasites that have very, very complex life cycles, such as the platyhelminths or flatworms that we discussed in other lectures. Parasites, the impact that they are going to have, which also we have discussed in other lectures, depends on the interactions that they are having with the specific organisms that they are infecting. When a parasite creates a symptom that is going to weaken the host is what we know as pathogens. We call them pathogens when they actually produce sickness in the organism that they are infecting. Okay? Not all parasites are pathogens, even though they are acquiring resources from the host that they are infecting. Okay? Questions? Now, positive interactions. So here the classical is mutualism, but also included here is commensalism. So mutualism, it comes in many different forms. Some of the most classical examples are fruit dispersal by frugivores because they consume the seeds, and the seeds have been adapted to pass through the gut of the organism in order to get rid of some layers that if they don't get rid of those layers and they drop to the floor, they are going to be inviolate. So the digestive system of the animal consuming the fruit and the seeds is needed in order to pre-process the seed and the seed being able to produce a small plant, okay? If that doesn't happen, then the seed is just going to be sitting there to be consumed by another organism. Other example of a mutualism is what butterflies and bees do for us, right? Pollination. So they are the ones doing all the pollination for the flowering plants that we have on the planet. And as we have said before, the most dominant group of plants on the planet are the gymnosperms. Right? Yeah, gymnosperm is the most dominant. No? Who's the most dominant one? Okay, good. You're still awake. That's good news. All right. Other example in the tropics is that between ants and plants. Usually, acacia trees are going to have what is called these little houses for ants called domatia. And they have these little thorns, or not that little thorns, that are like huts that have an opening, and that's the house for the ants. And the ants basically get food resources from the plant, and the exchange for the plant is that they are going to protect against herbivores. As soon as there is a herbivore trying to eat the acacia trees, the ants come in swarms attacking that herbivore. And also they clear competition for the acacia tree in terms of resources by clearing up a circumference around the tree, like in this picture here. This clearing here is not a natural, well, quote unquote, not a natural clearing, but rather a clearing made by the ants to get rid of some competition for underground resources for the plant. Now, of course, ecological interactions imply that there are trade-offs, costs and benefits. And when you are talking about a mutualism, in order for the mutualism to remain in place in nature, you need that the benefits to each of the partners engaging in the interaction are higher than the costs that they are paying. Because what happens in nature, as you by now know, the environment or nature is not stable enough. There are always changing conditions through time. And in some years, it's not going to be convenient for one of the partners to remain in a mutualistic interaction. For example, if a plant species that usually is engaged in mycorrhizae mutualistic interactions, because the mycorrhizae helps the plant to get more resources, mineral resources from the ground, suddenly this plant manages to disperse to a place where the soil is rich in nutrients. Well, maybe the cost of having the fungi in the root system is higher than the benefit, because now there is plenty of food resources in the ground for the plant, that there is really no benefit of having that partner. And that has been observed in nature. The same plant that usually engages in mycorrhizae interactions is going to get rid of the fungi partner in some places where the soils are rich. Okay? Commensalism is an example also of an ecological interaction that you cannot think of ecological interactions as set forever in that way. So it can be different. They can go from mutualistic to commensalism to competition to parasitic interactions, depending on the environmental conditions. So in commensalism, you have one species benefiting and the other really doesn't care. Like, for example, cattle egrets. Usually they go around the cattle because when the cattle is moving around the grasslands, they are creating this movement of the vegetation, so they are flushing insects, and basically it's easier for the egrets to catch those insects that are being flushed by the cattle. That's why they are very close to the cattle. But other times, actually, it becomes even a more intimate relationship, because the egrets have also managed to take the ectoparasites on the cattle as food. So many egrets, in addition to the insects that are flushed by the cattle, they jump onto the cattle and start eating some of the insects that are parasitizing the cattle, like ticks or mites. Okay? Now, interactions, in particular positive interactions, can have a strong impact on ecological communities. What it has been shown with some research is that, for example, when you have certain species present in an environment, they are going to increase the richness of the community compared when they are not there. Meaning, they do something in their ecological niche and their function in the environment that gives the opportunity to other species to arrive and colonize those areas and thrive on those areas. For example, this huncus species that is common in salt marshes, when it is present, the salt marshes plant species diversity or richness is going to significantly increase. When this huncus species is not present, the richness significantly declines in those salt marshes. We will talk more about this in a second. Are there any questions? More seconds. Okay, let's move now. We have covered so far the generalities and details of ecological interactions and why they are important. Now we are moving more at the level of the community in general and the diversity that is the species and the abundance of species that are in those communities. What we have to remember, and I have discussed this before with you, is that the species diversity is not the same as the species richness. Diversity is a composite measure of whatever is living in an environment. And the two parts of diversity is species richness, meaning a list of the species that you find in that locality, and the other component is the abundance of those species in those localities. How many individuals of each species that we recorded are present in that locality. This is going to be more helpful with a simple example. We have two communities of plants, and we have, in terms of proportions of the different species, equal proportions of the 4 species of plants present in this community. And then we have another community that also has the same 4 species of plants, but the proportions are very different. One of the species is highly dominant in the second community. 80% of the individuals in that community belong to the species A. Which one of these two communities do you think is going to be more diverse?
The first one.
The first one. Why?
Because there's a more even quantity of each species.
Say it again.
There's a more even quantity of each species.
Mm-hmm.
There's not one that's like much larger.
Yeah. There is a higher evenness, is how it is called in ecology, or more even quantity of different, in the abundances of the different species that you find in that locality. That quantity about even abundances across species in a community is what we call in ecology evenness. What is the evenness? If there is a high evenness, it means that the species in that community more or less have the same quantities or the same proportions. Okay, now let's move into some diversity, a diversity index that is commonly used to measure how diverse a community is. So that is the Shannon Diversity Index, which basically is just a measure where you take the proportion of every species in the community and you multiply it by the natural logarithm of that species. So this is the basic formula, and what you do, for example, with the figures that we show in the previous slide, for community 1, every species have the same proportion, a quarter or 25%. So what you do is just plug in that proportion into the formula, which is 0.25 times the natural logarithm of 0.25, multiply 4 times. Why 4 times? Because there are 4 species and every species has the same proportion. Now, for the second community, every species has a different, or two species have different proportions. So what you have here is the same formula just multiplying by the proportions of each species. 80% for the dominant, 10% for the less common species in the community, and two species that have 5% abundance in the community. And here are the numbers that you get, okay? This is a very simple, easy way to see that community one has higher diversity than the second community. Of course, this is just one very general index to measure diversity in a community in nature. There are other indices that stress other quantities. For example, Shannon diversity gives higher weight to rarer species compared to, for example, Simpson's diversity index, which gives higher weight to more common species. So everything depends on what you want to know about the community. And this is one way to study the community structure of a place. There are many other ways to analyze the community structure, okay? Now, one thing is that for sure, when you arrive to a locality, it's not an easy task to study the diversity of organisms in that place, because many organisms are not apparent, are not easy to find. Many of them are going to be cryptic or microscopic. So remember, we saw that most of the diversity of this planet is on what domain? What is the domain that dominates the life on this planet, or domains? How many domains do we have on the planet? 3. Which are? And what are the dominant ones? Bacteria and archaea. And are they apparent? You can see them with your eyes? No, right? So you need certain tools in order to be able to really tackle the question about diversity on the planet. So, what many people have done with recent sequencing technologies is take what is called environmental samples. Environmental samples means just basically that biologists go to the field and grab water samples or soil samples. They do some preparation and then just put them into the sequencer machine, and then they get out a lot of genetic sequences. Now the task is to identify what are the organisms represented in those genetic sequences. And sure enough, we have been very successful at identifying many new species of microorganisms using this type of samples. And that is allowing us to understand the diversity of microbial communities and understand how they change, how diversity of microbes change according to certain environmental factors. Because if you remember, environmental factors are the ones that are responsible for the distribution and abundance of species across the planet. What are the most important factors for terrestrial species? What abiotic factors are the most important ones affecting the distribution and abundance of terrestrial organisms?
Water, temperature.
Yeah?
Oh, wait.
Temperature and precipitation. So anything related to humidity is important, okay? So those two. And are those important in the aquatic environment? Are those two important in the aquatic environment?
Maybe temperature.
Temperature, yeah, of course.
Like saying, species in, say, Oregon are different than the species found in the waters of Florida. Like you won't find like a rainbow snapper somewhere in Washington compared to here.
Okay. So, and what are then the abiotic most important conditions in aquatic environments?
Light.
Yeah, light, for sure. Because you have two zones in aquatic environments that are called what? When you have light, that zone is called what? Versus the other zone where the light is no longer present? Yeah, photic and aphotic, okay? So there is light, no light, okay? And if there is no light, how do you have food webs in the place where there is no light? How come? There is no plant material, there is no food. How is food produced in those food webs?
Bacteria or small plankton.
Maybe.
Like dead organisms, like goes down into the ocean.
Okay, that's the detritus, yeah, that's a lot of food that sinks into the ocean, but there are very complex communities in the benthic portions of the ocean where there is no light. So what is other name, okay,
Is that the chemoautotrophs?
yes, chemoautotrophs, okay? So plants are light autotrophs, but the ones that don't get light but become the primary producers, meaning they take the role of plants in places where there is no light, are chemoautotrophs, okay? All right. So, and in this example, what researchers have found is that the pH in the soil has a strong effect on how the diversity of species of microbes changes. And they measure the Shannon diversity index, and as you can see, as the pH becomes more neutral, diversity increases. But there is a limit, right? As soon as it becomes more basic, species diversity of microbes starts to decline. Other important thing about the diversity of species in communities came out from this long-term study from decades of a person, a community ecologist named David Tillman, that did these large outdoor experiments where he created with his team all these different plots that you can see here in this field, and the different plots have different numbers of species. So they basically use the same set of species, but what they varied was the number of species per plot. And what they did was to measure many different attributes of those communities of small plots. And what they found was very enlightening in terms of how important diversity is. The more productive plots, the ones that produce the most plant material, were the most diverse ones, meaning the ones that have higher diversity, more species richness, and more evenness. Now, the ones that were able to withstand disturbances were those that were more diverse from year to year. So some years were colder than others, and the ones that had a higher diversity were better at recovering and reaching the former state. What other thing? Also, that is not mentioned in the textbook, but they plots that were more diverse were more resistant to pathogens and to invasive species, meaning those that pose potential competition. And the idea goes that usually when there is higher diversity, an invasive species is going to be facing a larger or more diverse array of what? Of potential competitors and potential predators. So they are going to be having a harder time trying to survive in those more diverse environments compared to simpler environments, which have been shown to be more susceptible to pathogens and more susceptible to invasive species. This, of course, leads us naturally to what we know as trophic structure. Trophic structure is just basically the feeding relationships that happen in a community, in a natural environment. Who is feeding on who? And that is basically by ecosystem ecologists measure in the transfer of energy from one level of the trophic structure to the next. And that put together is what form food chains. So in a food chain, you are going to have different trophic levels. With this picture, it's going to be easier. So These are different trophic levels of a food chain. Depending on what habitat you are studying on the planet, the species interacting are going to be different. This is going to be a terrestrial food chain. This is going to be an aquatic food chain. And the primary producers at the bottom are going to be plants, and over here, the phytoplankton. So those are primary producers because they can do photosynthesis, convert light energy into chemical energy that can be actually consumed by other organisms. So this is the most basal trophic level. Then you have primary consumers, which represent another trophic level. And we move on up the food chain to the top predators or top consumers in the food chain. Now, there are many different things that we will talk about in a second about food change. The first one is that usually when you go to nature, food change don't look like this. This is a simple representation just for you to understand trophic levels. Primary producers, primary consumers, secondary consumers, or carnivores, herbivores, etc. But food webs are more messy or messier. So it looks something like that. The circles are going to represent the species that you find in that food chain, okay? And the arrows are going to tell you who is getting benefited by each one of those organisms. As you can see here, the phytoplankton becomes the food of krill and also the food of copepods or the zooplankton. And the arrows pointing, you can see that many different organisms, like for example, this carnivorous plankton, that is like a primary type of consumer, is going to become food for many different species in this food web that are going to be secondary consumers. And these secondary consumers are going to be consuming many different levels. They are going to be consuming primary consumers and also primary producers, okay? And so you can see the complexity. So it's not, in real life, it's not easy to study the food webs and the ecological interactions in them. So what we do sometimes when we have a complex food web is to break it into smaller pieces. And those smaller pieces usually are the one, we select those that are less connected to other trophic levels. Because usually when you try to simplify a food web, you are also going to miss a lot of the ecological dynamics that are going to impinge into you understanding that sub-module of that food web. So we do it just for convenience, but after we understood the dynamics of this sub-web, we need to put it back into the larger food web to know how conditions are going to be changing between the different trophic levels. We will talk more about that in a second. Okay, let's take this question and then we can take a few minutes for you to stretch or go to the restroom.
these many. I could probably get some done. If they're not working. Okay, this would work sometimes. This one's only 250 words. 200? Okay, so basically it's only 500 words. This one basically 750 words. Yeah. And then these are some. Yeah, I could get some. Oh girl, I probably can't do that. Probably not. These are my figures. 800 words. Yeah.
No, we can do it. Because my advisor, services, they told me, yeah, I
Okay.
need 30 credits.
I know, it does boost your grade. Boost your grade so
Yeah, that's what I'm saying. Like, what?
I'm scared.
If I just click on the window, it counts as going out of the tab, but I didn't really do anything. And sometimes I just get a notification.
I'm like preparing my mental speech to be like, I didn't read totally.
Why did my kids have to be punished for the fault of half of the time? Like, why are we being, like, I might as well just not try for this exam, because at this point, actually, I'm just gonna take the time to study for final. I have like what, 4 different exams in the span of a week. I've never tried to my 6. Yeah. No, I genuinely wish that the semester was longer, actually. It went by way too much.
Okay. So, when we are talking about food webs or food chains or trophic levels, we know that there is a limit to how long those chains can be in nature. And the longest food chains don't go beyond 5 or other ones, I think. I don't know what the record is, but usually no more than 5 links or trophic levels. And one of the reasons for that is known as the energetic hypothesis. Basically, it states that there is a limit to the number of links that we can have in a food chain because there is inefficiency in the transmission of energy from one level to the next one. Okay? So because remember, plants capture sunlight, transform it into plant material that can be consumed by herbivores. But plants also are going to waste a lot of the energy that they receive. In what form? In the form of evapotranspiration or some waste material, etc., etc. So not all the material that has been converted from light to plant material is jumping into the next trophic level. So if you take that example over here, if you have plants that have 100 kilograms of plant material, if we take the energetic hypothesis that states that about only 10% of the energy in one trophic level is going to move into the next trophic level, then those 100 kilograms are only going to be able to sustain about 10 kilograms of herbivore material. And those 10 kilograms of herbivore material are going to be able to sustain only 1 kilogram of secondary consumers or carnivores, okay? So, because we are not that efficient at transferring energy from one trophic level to the next, that is what imposes the limit of how long a trophic chain can be. And it depends, of course, on the environment where you are studying. If you are in a highly productive environment, then the number of trophic links is going to be longer than if you are in an environment that is considered poor in terms of the resources that organisms can consume. And here is an example of what some researchers did in these tiny ecosystems of holes in tropical trees, where those holes, they can retain vegetation material, and that material becomes like the bottom of the food sources for many other organisms that are going to depend on those. So what they did is to do some removal of material. The control, sorry about that. The control basically is what it is the natural rate of litter fall into those tree holes, okay? And when you don't do anything, they can sustain trophic all the way to 5 trophic links in just those small ecosystems on a tree. But when you remove 1/10 of that litter material in the hole, then there is not enough energy. You are removing energy for that tiny ecosystem. Now the trophic links recorded are only 3. And if what remains of the litter material is 1/100 of the original amount, then you can only sustain a very, very small trophic web. Basically 1 link. Whoever is consuming the litter material that is in there, okay? Questions? Okay. So, within communities, we are going to have species that have more significant roles. There are species that have very large impacts. Some species that have large impacts are known as foundational species. And those foundational species are very important for the ecological dynamics, either because they are large in size, like trees, or they are highly abundant, or both. They are large and abundant. For example, a foundational species for a pine forest are pine species, okay? Pines are big and are abundant in that type of ecosystem, okay? That would be an example of a foundational species. Now, usually, but not always, foundational species are going to be dominant, and as dominant species, they are going to be competitively superior to other species. Yet, that only happens after certain ecological succession, and we will talk about ecological succession in a second. One way that people can measure the impact of those foundational species, you can guess, is by removing many of those organisms from the environment. And people haven't had to do that in an artificial way. People have recorded the impact of a foundational species due to epidemic of pathogens. For example, the oak blight that basically kill thousands of oak trees across the forest here in North America have a large impact on other species that depends on the forest, because basically the forest is the house for many other species like birds that nest on trees, etc., or the foods that are produced by those trees. So once you affect those foundational species, basically you are deteriorating a lot the community and the dynamics of that community. Other species that are very important and that have large effects are known as keystone species. And unlike foundational species, keystone species usually are not the most abundant species in a community. They can be top predators. And if they are top predators, we know by trophic interactions that you cannot have more top predators than primary producers, because there is not enough energy to sustain more predators at the top of the trophic chain than the producers that sustain the trophic chain, okay? So, keystone species, the most classical example in ecology is that one of that sea star, which is a predator of shell organisms. And the role of the sea star is to maintain a more diverse aquatic community. Here in the graph, what you have is years since an experiment started, and on the y-axis you have the number of species present in that community. And what you see is that when the sea star is present, the species richness is high. But when this top predator, the sea star, is removed, the species richness in that community plummets, and it remains fairly low. And why? Because the sea star controls the population of herbivores like sea orchids. What happens when there is no control on herbivores? They are going to decimate the community of plants. If you decimate the community of plants, what is going to happen to, for example, other organisms that need plants for food production? They are going somewhere else because there is no longer food. So richness is decreased. Okay, so that is an example of a keystone species. Other examples of keystone species that are also known as ecosystem engineers is the beavers. When they build their lodges, they basically create wetland environments, and that opens the opportunity for colonization of species that wouldn't be present otherwise, because conditions before the beavers arrive are less wet or drier, but when they construct their dams, then suddenly you have these water reservoirs that open opportunities for many species. If we are thinking about birds, aquatic birds are going to be colonizing those areas that formerly were dry. Now, some foundational species like trees, like the examples that we discussed, are also considered ecosystem engineers, because at the end of the day, we characterize ecosystems based on the dominant species of those ecosystems, okay? So in the case of the simple example, pine forest or pine oak forests, the names come from the dominant species. But if you move, for example, to a tropical forest, there is no single tree dominating the ecosystem. There are many species dominating the ecosystem, but the evenness increases, okay? But again, trees become ecosystem engineers. Now, when we are talking about the dynamics, the interactions between the different organisms, trophic webs or trophic change can be controlled in two different ways. One is called bottom-up, and the other one is top-down control. Bottom-up control is when the primary producers are the ones dictating how long the trophic chain is going to be in the environment. Why? Because if there is not enough food, there will be not enough energy to be transferred to other trophic levels in the chain. So bottom-up control is when that condition, primary producers are the main ones to sustain the trophic chain. Now, on top-down control, what happens then is that the control on the trophic interactions in the food web is going to be conducted by the top predators. And what we observe in trophic interactions is that when we remove some of the levels, we affect the positive and negative interactions in the indirect interactions that we observe in the community. What do I mean by that? Let's take, for example, here, this food chain, where we have the top predator, which is the orc, and this one is going to feed on otters. Otters are going to feed on sea orchis, which feed on the aquatic plants, okay? All these arrows represent basically positive interactions going up and negative interactions going down. But when you are looking at this, there are what we call indirect interactions. The orc is going to have a positive let's see, it's negative, yeah, a positive indirect interaction with the sea orchids, because it's going to be removing the predators of the sea orchids. At the same time, the otter is going to have a positive indirect interaction of plants, because it's basically going to be eating the sea orchids. If we remove this top predator, then the indirect interactions are going to be changed, because now this one is going to decimate this herbivore. And then this is going to have an impact on the community that is going to be overgrown. There will be a lot more plant material than used to be when this herbivore was present, okay? So that thing of changing how species affect each other in terms of positive and negative interactions, and what happens when you remove one of the levels in the trophic interaction, is called tritrophic interactions, okay? I'm going to let you watch this one on your own, because it's fairly long, but it's the story basically of how some ecologists came to recognize what keystone species are, using the example that we discussed, the sea stars. But I'm going to move into the other material, so we have time to finish this lecture. If we have more time at the end, I will come back to this video. Now, one thing of knowing the ecological interactions in a trophic food web is that we can use that in an applied fashion, okay? We can use our knowledge of the trophic interactions in order to control certain levels of the trophic chain that are creating some problems. For example, we have a lake, and when that lake was disturbed, what we have is that fish were fairly abundant, so plankton was rare. Of course, that was beneficial for algae, right? Because a lot of fish were eating a lot of zooplankton, so they had a positive indirect effect on the abundance of algae. And what is the problem when we have a lot of algae in lakes? Like, for example, right now, if you pay attention to the lakes and lagoons around here in Florida, there are algal blooms right now. You see the little carpet of green on the surface of the water. So, what happens when we have that to the aquatic community in terms of chemical composition and things that other organisms need? Well, now red tide is one example. That is more extreme. I'm just talking about eutrophication, the process of eutrophication. What happens to a water body when there is a lot of eutrophication?
So say, the red tide, that there's too much oxygen in the water, right? Or
There is less.
Oh, that's all.
Yeah, eventually. At the beginning, because there is more plant material, there is more oxygen. But eventually, if there is too much plant material, then it's going to be the opposite. Because then animals start to die, there is not much light reaching the bottom of the habitat. So all those are changing through time. So that reduces oxygen. With less oxygen, many species are going to die. So that's why people care about restoring what we call polluted water bodies, which is also eutrophication. So what they did is to reduce the number of fish, and by reducing the number of fish that were feeding on zooplankton, they allowed the zooplankton to produce higher abundances. More zooplankton means what? Less algae, because they are feeding on the algae. So in this case, what we did was to change the indirect interaction from a positive effect on algae abundance to a negative effect, because we reduced the pressure of fish on the zooplankton. So this led to a change from positive indirect interaction to a negative effect indirect interaction on algae. Okay? So again, having that knowledge of ecology and trophic interactions can be helpful in order to fix problems in the environment. More seconds. Okay. Questions? No? Many decades ago, in the middle of the last century, even earlier, many ecologists favored the idea that biological communities are at equilibrium, meaning they have reached a climax community structure unless they were heavily disturbed by certain activities like human activities. So they thought as competition had been a factor that leads to a stable community composition. So what they were thinking is that once we reach a stable climax community, that is it, that is how it's going to remain unless there is a very drastic disturbance, okay? But then another school of ecologists came over, and then they challenged this view of a single climax community, especially because we can have the same type of community of organisms in in different geographical locations, and conditions are going to be different. So soils are different, abiotic conditions are different, so you cannot expect a community of organisms to reach exactly the same position after many years. So rather what they said is that communities in nature, an ecosystem in nature, even though they can reach certain equilibrium, they are always under what they call dynamic equilibrium, because they are always suffering certain changes because we affect them with our activities, or there are natural disturbances that are going to affect the community composition. So they basically restart the assemblage of the community from different different time points. We will see several examples about this. So they were saying that communities are rather adapted to disturbances, and they can reach certain equilibrium. And what they the most favored hypothesis, and the ones that has the most support, is what is called the intermediate disturbance hypothesis. It means that the number of species in a community or an ecosystem is going to be higher when you have intermediate disturbances in the community, meaning it's not a community that presents no disturbance at all, like it was proposed earlier by some ecologists. Rather, what we observe in nature is communities that are regularly disturbed, and those communities that undergo regular disturbances, it doesn't have to be a drastic disturbance, are those that are going to have a higher species richness. And that is what is shown in this graph. You can see here the disturbance index. When there is very little disturbance, the number of taxa present in that community is going to be very low, which is what we ecologists call a climax community. A climax because it hasn't been disturbed for so long that you have the same species dominating that community, and no other species can enter that community because they haven't had any chance to colonize that community, okay? But as you start to move across the index of disturbance, with higher disturbance, you open opportunities, other niches for other species to arrive to the community. So the number of species starts to increase. But if disturbance is very large, like for example a hurricane or humans destroying or altering the environment, then that is going to reduce again the number of species located in that community. So it's a home-shaped curve what we will observe in nature. So drastic disturbance events are going to lead you to poor communities. And climax communities, on the other extreme, when there is very little disturbance, also lead you to poor species communities. The most species-rich communities are those that undergo regular disturbances. Here we have some examples that have occurred in nature, especially in seasonal environments. So what you have here is a community of forests, pine forests, that are adapted to seasonal fires. And when the fire arrives, of course, you can see here all the opening, the community goes down to almost zero, right? It's barren. But then the successional process starts. The successional process means that the species start to colonize again. But in this case, the first ones to colonize again are the species that are adapted to fire and that need seasonal fires to occur. And this is 1 year after the fire. You see now that plants are coming back, and when plants are coming back, other organisms are going to start arriving into those areas again. Questions? More seconds. Okay. Okay, so this, what I've been discussing, is what leads us to the concept of ecological succession. Ecological succession is what the name implies. There is a succession of organisms across time in nature, okay? Now, there are two types of succession: primary succession and secondary succession. Primary succession is the one that we discussed when there is a large disturbance that creates a really different landscape to what was there previously. One example is what has happened in northern latitudes with icebergs. With the retreat of the ice caps, many things have changed. Let's take this example in Alaska. So this was the glacier back in time, okay? But the glacier started to retreat in the year that shows up in here. So since the glacier started to retreat very long ago in here, you start to see a community that resembles something more like a climax community, something that is already in later successional stages. But when you go to the more recent portions where the ice cap has just recently retreated, what you see is what? Just barren land with a few pioneering species, okay? And these pioneering species are important because what they are going to do is to change the chemical composition of the soil. What is barren rock is going to be diluted into more available nutrients for other plant species that are not able to colonize initially. So this second stage over here, which is known as the Dryas stage, you can see now more plants covering this stage. But the reason these plants were able to colonize that area is because of the changes that pioneering species made to the soils in that environment. Then after this one, we move to the third stage where you have other tree species, okay? And then eventually, the later stage in this portion of Alaska is going to be a spruce forest, okay? That is what we mean by primary succession. Also primary succession you can see after a volcanic eruption. Like for example, Mount St. Helens, when it exploded, all around it was barren, and little by little, the ecological succession started to occur. First with the pioneer species, then the next set of species, and with the complexity of later stages come other species that are able to colonize. So in this graph, basically is what I just explained. But now, taking as a reference nitrogen, which is one of the most important nutrients for plants in the soil. As soon as the nitrogen starts to increase, you start to see that there is a replacement from the pioneers to the subsequent species until we reach the spruce stage, okay? Now, secondary succession is when there is some disturbance in the environment, but it's not as drastic as the ones that we just mentioned, okay? For example, if we use some portion of the land to have maize, but then we abandon that land, I mean, it is still green, right? There is still plant material, but then eventually it's going to be colonized by the native plants in the surrounding forest, surrounding that, that land that was filled with maize initially. So when we abandon crops, usually nature takes over. So that is what an example of secondary succession. Now, in terms of the strongest disturbances, sadly, we are the ones that produce the strongest disturbances due to our activities, and in marine environments, you can see the classical example is what we do with, when we are basically trawling the bottom of the ocean, trying to capture all these shell organisms and many fishes. This is how it looks before we do a trawling event. And this same community, this is how it looks after we finish trawling that environment. So basically it is barren, there is nothing left in there. So of course we've been trying, or conservation biologists have been trying to improve the techniques that we use in order to exploit the resources of the ocean. And there are some improvements. Okay. That is in terms of ecological succession and disturbances. Are there any questions? No? Okay. So let's move a little bit more into biogeography. We have talked about biogeography earlier, but let's do a little bit more in terms of what happens. So I mentioned something that is very important in terms of species diversity, where we know that species are going to be richer where on the planet? in the equator, okay? And anybody remembers what is the name of the theory stating that richness that is larger in the equator versus more northern or southern latitudes, meaning moving into the poles? No? Okay. It's called latitudinal species gradient. Okay? So that is one portion that we have discussed. The other portion is in relation to the area that is available to organisms. Where do we expect to find higher richness? In what places? When we are considering area. Yeah, tropical areas, but when we are just talking about the area, not the latitude. When you are comparing sizes of places, yeah, larger areas. So the larger the area, the more accumulation of a species you are going to have. Basically, what is behind that is because there will be more niches to exploit compared to a smaller area, like small islands versus large islands. If you compare, for example, the Hawaiian Islands, tiny islands like, I don't know, Middle Island in Midway Island in Hawaii, it's tiny, right? If you compare that to the main island, Hawaii, you are going to see a lot more diversity. Why? Because there are a lot more niches that can be exploited by different species. So this basically is what it summarizes latitude and area. Species richness is larger, closer to the equator, and is going to decrease going towards the poles. Larger the area, more species, okay? Now, two main ideas behind the latitudinal species gradient are going to be evolutionary history and climate, okay? They are not mutually exclusive. So what biologists and ecologists think in terms of why we find more species in the equator has to do with the history of those areas, especially in relation to large disturbances. The last set of disturbances that occur after the impact of the meteorites 65 million years ago was the ice ages. Those increases of ice caps and retreating of the ice caps. And where do you think animals were surviving when the ice ages were at their peak? Closer to the equator. They were moving closer to the equator because the equator didn't have that big of an influence from the ice caps. The ice caps affected northern and southern latitudes, okay? So climate was more stable and less disturbed for a very long time, millions of years, in tropical areas. So that allows what? To evolution. When you have more stable conditions for very long time, that allows evolution to produce novel adaptations, and with that the exploitation of new niches, and with that the accumulation of more species, okay? So that is one of the hypotheses behind why in the equator or tropical latitudes you have a higher diversity of species. Now, the other one in terms of abiotic conditions, as we mentioned, is climate. And when we learn about the sunlight radiation, we learn that radiation is heavier on tropical areas. They receive higher amounts of radiation in the tropics because the angle that the light arrives on the tropics is basically direct. There is not an oblique angle of the solar radiation arriving to the planet. That occurs toward the poles. So there is less radiation, less energy arriving into the poles compared to the tropics. So imagine what we just discussed about trophic interactions and the length of trophic chains. To have longer trophic chains, we need what? more resources, more biomass, more energy, right? Where are we receiving more energy in the planet?
I was gonna say areas that receive more sunlight.
Which is the tropics, right? Which is the equator. So more energy, more potential for higher biomass. Higher biomass gives the opportunity to have longer food webs or more trophic levels, okay? So that's another reason why we believe the tropics are richer in terms of species or more diverse compared to other areas. Now, one other feature that is very important to understand how we measure richness of different groups, it has to do with the production of water vapor by plants, which are the primary producers. Evapotranspiration is a very important measure of how productive an environment is and where we are expecting to find a higher diversity of many different organisms, not only plants. For example, in this graph, what we have is that an evapotranspiration index that we can measure from space. And as the evapotranspiration increases, you can see that the vertebrate species richness increases up to a certain limit, okay? So more evapotranspiration, it means larger green surface, which means more food, more biomass for everyone. And that is going to transfer across the trophic levels in the community. Area effects, we have already talked about area effects I think 2 lectures ago. The larger the area, the higher the species numbers that we are expecting to see. And these together, area with distance from source, from different sources of colonies, is what leads us to the island equilibrium model that we reviewed couple lectures ago. Meaning that how being larger or smaller and closer or farther away from a source is going to affect the number of species that you are going to find in those locations. And that, if you remember the video that we watched a couple lectures ago, shows that we have to consider on the x-axis the number of species that we find. The example is on an island, but it can be also on mainlands, different fragment sizes on a mainland environment. And then we have extinction rates and then immigration rates, okay? So where these 2 lines cross is going to be the equilibrium number of the species or the species number that you expect to observe in that locality. Now, if you remember, when we have high immigration rates, what we are going to start to see is that there is a lot of arrival of species. And then, because there are no much competition, immigration rate is very high. So a lot of organisms are arriving, okay? But as soon as more organisms start to arrive into the community, then competition starts to kick in. Because now more species are competing for resources. So the immigration rate starts to decline. Of course, it is declining, but the number of species starts to increase. But at the same time that the niche or the environment is filling up, more competition, more predation, etc., extinction rate starts to increase, okay? Why? Because now there is less resources and a lot of competition going on. So when there is a threshold number of species in an area, and you go above the level of this equilibrium point, extinction rate is going to be higher than immigration rate. And eventually you are going to settle in this equilibrium point that is going to give you the more or less number expected of species in that locality. Now, where this equilibrium point is going to sit in this graph is going to depend of the size of the place, the size of the island, and how close or how far away the island is from the source of colonies, okay? I'm not going into more detail now because we already saw one of the videos in the first lecture on ecology. Now, this theory was put into test by an ecologist known as Dan Simberlock. What he did here in the Florida Keys was to basically hire some contractors that do fumigation work. And what they did was basically to spray mangrove islands and cover them with huge plastic tents, okay? So they would kill every single insect on those tiny mangrove islands, but the mangrove islands were selected for different sizes and different distances from the mainland or from the source, meaning mainland Florida, okay? So they recreated what was expected in the theory. And sure enough, they found the results that were expected by the theory. The closer and larger mangrove islands have the higher species richness compared to smaller and farther away mangrove islands, okay? So after they kill all the insects, they started to see the recovery of the insect community. They started to see how the insect community started to come back. And then they observed the predictions of the island theory of biogeography. But one thing that was a little bit unexpected for them is that the community composition of insects was not exactly the same that was before the insecticide disturbance. So meaning, in terms of just numbers of species, the communities recover to former standards, the community before the insecticide, just in terms of numbers. But when they look at the identity or the composition of the species, they observe many changes. Not all the species that were there before were present after the fumigation experiment. So that's another thing that creates certain stochasticity in ecological communities. Okay, so that the environments recover doesn't mean that every single species that was there before is going to be able to recolonize those areas. This is the video that we watched a couple of weeks ago, so I'm not going to stop in there. You can watch it again later on in preparation for the exam. Now, pathogens. We've been talking about already on pathogens and the problems that they can create. And they can be strong forces in how communities are structured, both locally and globally. And we already talked about some examples. One example that is very dramatic is in amphibians. Anybody remembers what is the problem with amphibians? What is happening to amphibians?
Oh, the fungus.
Yeah, anybody remembers what fungus?
Is it titrates? I can't pronounce it.
Chytrid. So it's a chytrid fungus that damages the skin of amphibians, and it's been behind, along with other factors like habitat destruction and habitat pollution, have been responsible for the extinction of several species of amphibians and the decline of several populations of amphibians. Okay, so that is one example. The other is the blight that has killed many oak trees in North America, and that has indirectly made a decrease in fiber species that were highly adapted to that type of ecosystem. So again, there are many examples of how pathogens can create big troubles to many other species in the community. Now, when we took the lens into humans, what we've been facing in the last 5 decades is a constant increase on epidemics and pandemics. Pandemics are becoming more and more common, and pandemics, especially the most recent ones, are known as zoonotic pathogens. Zoonotic pathogens because the things that are jumping into human populations come from other animals. and they can be either directly transmitted, like the coronaviruses, the COVID that we just suffered a few years ago, or they can be vector-borne, and we have given examples of vector-borne diseases. So, these are things that are serious, and the problem with that is that in the case of human pathogens affecting us globally, we are responsible for those things, because as we destroy the environment or we modify the environment, we start to change the ecological interactions. And organisms that formerly were not interacting with humans now are becoming more and more common, or host switching into humans, because we are giving the opportunity for that. And also because when we alter the environment, sometimes we affect the regular host of a pathogen. And since we become the most abundant organism in those environments where we are living, we are an easy target, so easier to jump from some species into humans, which are fairly common in the novel conditions after the disturbance of that environment. Now, this is an example that we have mentioned also, the Lyme disease. The importance of identifying who are actually the responsible reservoirs for holding most of the infections in the community. Because if we know what species are the most prevalent and the most infectious ones to transmit that Lyme disease into other species, then we can manage to control that pathogen into jumping into other species. Are there any questions? No? Okay, 10 more seconds. All right, this is just a summary slide, but you are ready to leave, so just go. Good luck next week on your exams. Yes? No. I mean, you need to understand the equation. But you are not going to be asked to, and if you need to do something, it's going to be simple things.
Okay, so you're talking about math formula, like, know where they are.
Yeah, probably.